everyone, I'm Firyat Mishra. Today I am going to explain about cells. So there are mainly two kinds of cells, animal cells and plant cells. Other fungi cells and these monera cells come in bacteria cells like amoeba or yeast. So let's first understand by our animal cell. So we humans are one kind of social animals. So in our body there are many cells, tissues. So this is the simple structure of our body cell. So this is huge structure. There, the outer covering of the structure is known as the cell membrane, and one fluid is filled in this cell. So what is that fluid? Is that empty? No, that is fluid called cytoplasm. After that, in the center, just like I am a human, walking, talking, doing something by the order of my brain. So like us, cells also have a brain, which is known as nucleus. Nucleus is called the brain of our cell because it controls most things of our cell. After that, what are the zigzag structure? That are the endoplasmic reticulum. In short, it's known as ER. There are mainly two kinds of endoplasmic reticulum. First is smooth endoplasmic reticulum. Second is rough endoplasmic reticulum. In smooth endoplasmic reticulum, no ribosomes are present, and in rough reticulum, there are ribosomes present as directed. Some first smooth will be there, then after that rough will be there. After that comes the Golgi body. Golgi body is a body located near our nucleus. In mostly RBC cells, Golgi body is not present. After that comes the centrioles. Centrioles help in the cell division. Just like I am a cell. Suppose I am a cell or I have two cells. So if I will not grow, then I will not grow. If my cell will not divide themselves, then I will not grow. So for the division of cell, this centriole help in the division. Just like there are two cells in my body, so it will divide and become 4, divide and become 8, divide and become 16, and more and on. After that comes the lysosomes. Lysosomes are also known as suicide bags of our animal cells. They are not present in plant cells. So what are the function of suicide bags or lysosomes? Lysosome, just example, I am dukhi, I am sad, sure of feeling. Then I will go and murder myself. That's known as suicide. So if a cell is injured or is bekar, therefore the suicide bags help in cutting them, and the enzymes help in making them pure, or they just finish them. After that comes the ribosomes. Ribosomes are the small bodies present in our cell. It makes rough and smooth endoplasmic reticulum. So after that comes our plant cells. So in plant cells, the outermost covering is known as cell wall. So just for example, there is a tree in front of you. First there is a bark, then after that pure wood is there. So in this cell, there like a bark, there is a cell wall which help and make cell rigid and give it a shape, just like rectangular shape or any other shape of the cell. So here mitochondria is present, and here also mitochondria is present. What are mitochondria? Just like I am walking. Without food can I walk? Without food, how will I get energy? So like this, mitochondria is known as the powerhouse of our cell. It provides power to the different cells and cell organelles. After that, mitochondria, lysosomes are not present here. So what is this big structure? That's a vacuole. Just if I will eat something, so I will excrete my waste products. So cell cannot, in plant cell, they cannot secrete their waste products. In animal cell, they transfer to another cell or organ. That is simply known as organ. This cell will execute its product by two different cells, and then different cell will continue to different cell. But in plant, there is nothing. So therefore, one vacuole is there, which contain its extra products that are wastes and many things. After that, there is also nucleus in center. So an ER or endoplasmic reticulum, smooth or rough. And ribosomes are also present. And there, one more kind. Cell wall is present, but cell membrane is also present. And if animal cell have vacuole, then they are smaller in size but many in numbers. But if not are there, so here big in large, but usually one in number. And then they are who discovered this? How we know that this is our cell, this is our cell? We use a different thing, different device, which is known as microscope. Robert Hooke, who? Robert Hooke was the first to see plant cells. He took a piece of bark and put under his microscope. Then it was years ago, about 200 years ago. So they uses a microscope called light microscope because they see the things using light. So by his device, light microscope. So by light microscope, he saw that in the bark there are some hexagonal structures. Then many hexagonal, like a bee house. Therefore, he was the first observer of cell. Thank you. I hope you like my video.